Hola, buenas tardes. Bueno, me toca presentar ahora a Nate, que va a compartir con nosotros su trabajo en macrosistemática de mamíferos. Realmente para mí es un gusto poder hacer esta presentación. Nate es profesor asistente y curador asociado de mamíferos en la Facultad de Ciencias de la Vida de la Universidad Estatal de Arizona, e investigador asociado de la Universidad de Yale y el Field Museum of Natural History de Chicago. Entre otras cosas, Nate estudia la evolución, ecología y biodiversidad desde una perspectiva espacial y temporal, integrando datos fósiles, genéticos y rasgos de vidas de las especies. El objetivo de su trabajo es investigar cuándo y dónde se originaron los grupos de especies, a qué ritmo evolutivo y en qué contextos de interacciones ecológicas. Mucho de su trabajo se ha centrado en la evolución de los mamíferos, con énfasis de roedores tropicales y de desiertos. Así que bueno, sin mucha más presentación, los dejo entonces con Nate y con su charla.
Ok, gracias Pablo. Wow, y gracias a todos. Boa. Mi castellano es un poco viejo, mas eu falo português também e mais difícil para falar em espanhol agora. So the talk will be in English, and I hope that is okay. The talk is being recorded too, and you can go back to it and slow it down and things like that. Bueno, same. So yes, today I'm going to be talking kind of in an overview context about mammal evolution and ecology and taxonomy, but I want to kind of start at the beginning, which is sort of full circle in a nice way, that the Congreso, this Jornadas event, was a joint meeting with the IMC in 2009, and I met many of you then in Mendoza. Fantastic meeting. Here got to see 𝒂𝒎💂💂 with my PhD advisor, Bruce Patterson. And really that was sort of the opening for me of learning more about Argentine paleontology and rodent diversity. And so I ended up doing my PhD work on Octodontoidea, this superfamily of caviomorph rodents, which many of you know well, spanning from 𝒐𝐚-𝒂𝒐𝒂𝒍 to ecomyid rodents, 𝒐𝐚-𝐭𝐡𝐚𝐧, 𝒔𝒑𝒆𝒄𝒊𝒆𝒔, and has this kind of amazing chromosomal diversity from 10 in 𝒕𝒏𝒏𝒐𝐚𝐬s all the way up to 118 in 𝒑𝒐𝒐𝒈𝒚 bulliviensis, and this really fascinating rodent, Tympanoctomys beareri, shown here, 𝒔𝒑𝒆𝒄𝒊𝒆𝒔 rats, right, that have not only a large diploid number of 102, but a very large genome. And so this kind of led me on an interesting chase during my my first postdoc working at McMaster, and with Ricardo and Agustina Ojeda, who I saw are here. Bienvenidos. Good to see you, or be part of some event with you here. I'm going to briefly talk about this work because for those that don't know about it, I think there's a lot of continuing open questions that are worth thinking about more. And so Tympanoctomys is way out as an outlier in mammal genome sizes, 8.4 picograms. And, given that humans are about 3.3 and the mean for all mammals that we know about is about 3, this is very, very strange. And then if you kind of zoom in to rodents, continues to be strange. And of course, Octodontidae. So there was some sort of an event evolutionarily couched that led to a big genome expansion in the Tympanoctomys and a few other species of Viscacha rat. And so this is the sort of phylogenetic context here, showing I guess 𝒂𝒎💂💂💧💧 is now 𝒂𝒏💏💏💧💧🧐. This is all one genus, but about a 5 million year divergence from 𝐞-𝐣𝐚𝐦 𝐼max, which has both a normal karyotype and a normal genome size. And so there's this sort of question about, was it a whole genome duplication, a tetraploidy event that led to that group having an expanded genome, or was it repetitive DNA, some sort of repetitive elements? And this sort of led us to the Monte Desert of Mendoza to collect animals that we could preserve with really high quality tissue and do some genomic work to try and investigate this. And so 𝒂𝒏💏💏🧧 habitat shown here, Salar de Añelo in Mendoza, and 𝐞-𝐣𝐚𝐦 habitat shown here near 𝐸𝒂𝒎💏, a little bit outside the park in San Juan, where we captured 𝐞-𝐼𝐚𝐦s. Very interesting, different habitats, 𝐞-𝐼𝐚𝐦s being more similar ecologically to Abracoma or to Neotoma in North America, and 𝒂𝒏💏💏🧧 being more similar ecologically to like a Dipodomys microps in North America. And we were able to collect different tissue types and do transcriptomics on these different tissues, as well as whole genome sequencing, kind of in a shotgun light coverage manner. And then we compared that to what was the expectation from a known diploid tetraploid pair in Xenopus laevis, the African frog. And kind of, I'll just go through one of these results because I think it's sort of telling, is that just looking at the whole genome sequencing reads and looking at these 35 MER 35 base pairs that are similar to each other, we see that most of these are not, there's not very many repeats of 35 base pairs, right? So there's like many small repeats, but then there's in 𝐞-𝐼𝐚𝐦s 𝐼max about 5.7 million are on these like kind of longer repeats, but then compared to 𝒂𝒏💏💏🧧 𝑏𝒐𝒏💏💏🧏, lots more repeats. Lots more repetitive DNA was recovered in the genome. And this coupled with the lack of finding expressed duplicate genes really sort of led us to conclude that the evidence that we have so far does not support whole genome duplication, kind of in contrast to the previous thinking. But we left a lot of open questions in this context. So is the genome expansion still ongoing, and why, and what kind of would have caused it initially anyway? And really needing to dig deeper into the genomic work there to understand what this mechanism would be. How could this possibly be adaptive, or was it the result of inbreeding or something else? And in the context of these ecological traits, particularly these strange bristles on the roof of the mouth, you can see here, and the enlarged renal medulla, does the genome expansion play any adaptive role, in fact? So I just kind of want to frame that as an interesting question that, if we zoom out and then we just sort of think about, okay, what in general, what are the factors that govern genome architecture in the context of chromosomal number, genome size, and then obviously things more like gene expression and gene function. And this type of work really got me thinking about gene flow versus isolation in a historical evolutionary context, and how certain traits may be related to dispersal ability, or that would then lead into population size differences, inbreeding differences, could end up having a long-term impact on a genome. And so then, zooming way out and thinking about, all mammal diversity, both at the order level and species level, and kind of comparing these patterns. These taxonomic orders are somewhat arbitrary, but they are things that we see as major groupings of mammals. But the species diversity is really unevenly distributed, and we know this, right? Most mammals are bats or rodents, about 2/3 of all mammals, in fact. And, you know, so this sort of question of, you know, why are rodents so species rich? Like what is it about rodents ecologically that that kind of drove this species diversification? But really that's the same question as asking, you know, why are some other lineages so species poor, right? So we have the aardvark as this, you know, one species, very old order, about 80 million years. And we know that there's fossil diversity along this branch, but in the present day, one extant species, right? So this ends up being sort of the same question of, you know, why is there uneven species richness across the tree of life? And that in turn could be framed as sort of, you know, why is there differential diversification in any case, right? So phylogenetically, some clades have more species than others, but then we also see this pattern in space, right? In the latitudinal diversity gradient, where we have some regions, especially tropical regions, have more species diversity than temperate regions. This is a well-known pattern, obviously not only just in mammals, but it definitely manifests in mammals. And sort of this question, you know, are these actually the same pattern, or is one kind of causing the other? Is it like, what is the relationship here? And sort of in general, you know, people have thrown around a lot of hypotheses in this context. We have sort of these adaptive stories about key innovations. So in the case of rodents, we have the ever-growing bottom and top incisors that are sort of self-sharpening and chiseling, that may have, you know, enabled access to different ecological niches. Conversely, there's this idea about just some clades are older, right? We have, you know, certainly differences in clade age, but also ages of different regions. So the tropics being older and historically larger than they are today, and temperate and boreal regions being fairly young geologically. And then there's this third idea, which is one that I have been kind of favoring, and I'll talk about this in more detail, is this idea of differences in gene flow and isolation, and that species traits are related to those factors, ultimately leading to differences not only in speciation rate, but in extinction rate, and that a lot of the patterns that we see across the tree of life are actually driven by high turnover and are somewhat ephemeral, but will continue to kind of cycle. And this is sort of part of the phenomena that we need to be aware of. So this idea of ephemeral species is one that was sort of first, maybe not the first people to think about it, but to like frame it in this way. A really nice review paper by Erica Brie Rosenblum and colleagues in 2012, where this idea that we draw our phylogenies with species tips, there's all these populations and individuals ultimately inside of that, and kind of divergence processes and coming back together and going extinct. That's happening all the time, and in some groups more than others. So that's sort of the main framework, and then I'll talk about this in sort of three main parts, thinking about mammal species, and in particular the evidence that we have for what a species is, thinking about mammal evolution, this new mammal phylogeny that we built at the species level, and talk about how we constructed that tree, and then how we can use that information and the evolutionary rates that come from it to study the ecology of speciation, which is, I think, kind of the main point of a lot of this work. And I better drink some mate, as long as I'm like an honorary Argentine right now. In fact, I got addicted to mate in 2009 on that trip, so I have you all to thank. It has enriched my life. So anyhow, yeah, so if you have not seen this photo, this is thought to be the first selfie of a mammal taking a picture of himself, a macaque shown here. And this question of, yes, what are species? I don't necessarily mean to get into a species concept type debate, but really whoever's defining the species, like whose taxonomy are we talking about? And, is this taxonomic perspective consistently applied across your analysis? Sort of the important thing. So sort of maybe issue one. And then of course, what about all the extinctions, right? So we know that the species that we see today have these long evolutionary histories, and there's been extinctions all along the way. How does that information or the lack of it influence our results? So of course, mammals are this kind of fantastic group to be studying in this context. I don't need to tell you all that, just that, especially in Argentina being such a rich fossiliferous environment, we get all this additional information. So these three fossils shown here are actually from the Mezzel pit in Germany, these complete body fossils that are pretty outstanding, but all the isolated molars and, partial dentaries and crania are additionally very helpful. And so phylogeny is often thought of as tree of life, but we really need to start talking about it as the tree of life and death, right? And a lot of this will come in when we continue to add extinct taxa into our analyses. This is a simulation shown here that in order to get to 25 extant species at the present, there's been a lot of extinction along the way, all these yellow symbols, right? So 80 extinctions in this kind of high turnover simulation scenario. But then we reconstruct it at the present in the context of just those 25 species, and we get a very different picture of the evolutionary rates, evolutionary history. So there's a lot going on these long branches here that we miss in the context of extant only phylogenetics. This is well known, like people have known this for a long time. We've tried to sort of ignore it in different ways and to get around it. But I think especially now people are realizing that there's a lot of bias if we don't really incorporate all these fossils along the long branches. And so there's some work that we've been doing recently to synthesize durations, kind of first and last occurrence for mammal genera, all the way across crown mammalia using public databases. And I'll talk more about this work a little bit later. Taxonomy is this other, extremely important component to trying to understand species, and that's because it's literally the science of how we define our species. What do we think is this kind of important discontinuous unit, and defining them by common ancestry and degree of divergence is the standard practice at this point. And there's this this really important point that the same researchers can see the same group of organisms and have different opinions or have different evidence that can revise a previous opinion, right? So we have a split in this context from 1 to 3 species. This is happening all the time, including by many of you in this room. A lot of great taxonomy happening in Argentina. Integrative taxonomy, multiple lines of evidence. It's outstanding. The thing is that we can't, we can keep track of this on a small scale, but on the sort of, major synthetic scale of, 2 million described species globally, that's across all taxa, but even just, 6,000 species of mammals, we need other methods. We need to be a little bit more, can't just be doing everything manually all the time. And so there's this, sort of core taxonomic problem of according to who and when, how do we know what we think we know? And that ultimately manifests because taxonomy is the language with which we communicate about biodiversity, right? We index all of our specimens and observations about those specimens, and then kind of species level insights by the species names. And that's great. It's just we really need to put about high value on taxonomy in that context. And in the same manner, taxonomy is then the key to understanding species-species interactions, or species environment, or species pathogen interactions, right? So sort of the key to unlocking this ecological knowledge, and it needs to be treated with again that that level of importance. So we can kind of, I say taxonomy, and maybe some people's eyes glaze over because they don't want to worry about taxonomy. Species, maybe they want to worry about because they're interested in conservation and things like that, right? Ultimately, we're talking about, you know, the currency, both in an evolutionary context of trying to understand the evolutionary past, and in the ecological present of trying to, you know, understand these interactions. So shown here is like a, you know, nice Sonoran desert scene from Arizona, where I am now, of, you know, we want to understand like who eats what, who competes with who has which pathogen. We need to know this type of information about the taxonomy. And this has come into kind of, you know, very fine relief for a lot of us in the last 2 years in the context of the pandemic, right? We have humans, wild mammals, viruses, and this kind of like interchange that's now heavily influencing our lives. But to a large part, viruses have been studied by virologists, you know, with some exceptions of some good mammalogists that have been, you know, taking vouchers along with the, you know, the symbiotypes along with the viruses. But so there's like an important role here for mammalogists to step in and say, okay, we can tell you about these host reservoir organisms, their evolutionary history, where the specimens are deposited, how to trace all this back into the taxonomy. But we all need to work together in this context. And so we had a recent sort of perspective piece in the Lancet Planetary Health that kind of puts forward this idea that, you know, we actually know a lot. A lot of it's locked up, what we're calling dark data, in books. It's like known, it's been written down, it's in, or maybe it's locked up in a PDF, but it's unstructured, or even if it is in a database, it's disconnected. And so it's not sort of explicitly like, this virus is a host of this, hosted by this animal. And with this evidence, you know, this PCR test was done on this gene. That's how we know. That's like our level of confidence. And ultimately, we want to get to this kind of broader ecological knowledge framework. I would broaden that out even more and say that, yes, we want to know the ecology of given mammals, but on the same, you have to flip it on the same way. Like, how do we even know what that mammal species is, right? So, okay, what's the geographic range map associated with it? Kind of the taxonomic concept, you know, it could be either a widespread concept or a narrowly defined one of the same name. Exemplar DNA sequences that have been vetted, that are, you know, not contamination, that are not mislabeled in GenBank. Type specimens, where are they? You know, like, they're in museums all over the world. Maybe, you know, a lot of species don't have types too. This is information that we need to kind of systematically go through and figure out. And ultimately, you know, some types have gene sequence from them. It'd be great if all types did, right? So this is, you know, a longer term sort of framework, but we need to be able to connect all this information. Just a quick example of really interesting case study in Madagascar of these mouse lemurs that were 2 species in 1993, but are now considered to be 25 species, right? And there's been a lot of, you know, systematic work, surveys, and molecular phylogenetic species delimitation approaches that have been leveraged against this. So it's been closely studied, but the interesting thing is that a lot of the same names have different taxonomic concepts associated with them, not surprisingly, right? So Rufus used to be this really widespread East Coast Madagascar species in 2008, you know, not that long ago, and then now it's just narrowly defined to this one part of the mountains. Similarly with Myoxinus and Danfossi or Danfosorum, kind of depending, also changes in the nomenclature. Things that we need to keep a closer eye on, and if we want to do any kind of systematic, or sorry, synthetic understanding across mammals. And we know this, right? We know that species are continuing to be described. There's been lots of splitting in the context of, you know, the phylogenetic species concept, and, you know, these are positive developments. But it's not just the number of species; it's all these rearrangements that are important. So our work on the mammal diversity database has been bringing this up into a current, like, sort of bleeding edge perspective. and shown that there's this, big 55% increase in described species since 1982, since Mammal Species of the World 1. But, similarly, even since 2004, there's about a 25% increase in species, and then all this rearrangements as well, right? And so we had this paper in 2018 that kind of introduced the mammal diversity database and documents a lot of these changes. That ended up really being the tip of the iceberg. Like, a lot of the work we've been doing more recently has been bringing in all the synonym information and the type specimens, type localities, and getting a better handle on a lot of these rearrangements. So yeah, if you haven't been on the website yet, go check it out, mammaldiversity.org. So our effort is sponsored by the American Society of Mammalogists and is, ultimately just trying to be an objective perspective on all the nice taxonomic work that folks in Argentina and around the world are doing in the context of, this science of taxonomy, right? And so, yeah, we're now up to 1.7 in our versioning. We have this core group of student research assistants that's been helping, but also we have a broader biodiversity committee at the ASM that has been, assisting with a lot of this work. And so if you're interested in getting involved through that context, you should contact me. So yeah, taxonomy matters, right? I guess I beat that drum, right? And I think we all need to beat that drum a little bit. And it's a little bit obvious, right? So it's like this meme here of, desafío. It's a little bit of a challenge, It's actually not. The capybara is very easy to see, and we should all put that forward. And that extinction events matter in the context of trying to understand evolutionary history. These things are obvious but need to be emphasized. So then thinking about mammal evolution, right, and from the extant perspective and from the fossil perspective. We had this paper come out in 2019, and where we published the phylogeny on the website here at virtlife.org. I'll talk a little bit about the way that we built the tree and the data that underlies it. So this is all public sequence data. This is all from NCBI. It's all data from about 2015 and earlier. So that's also important to consider if you ever are wondering, like, oh, why isn't there data for like newer published data in the tree? We need to kind of repeat the analysis and reconstruct the tree to do that, but we should. Basically, the way this data was assembled was not trusting the labels on the genes or the taxa, really, which was to kind of go in. We had a curated sequence or curated set of sequences, these bait sets, one sequence per family, that we could then blast and retrieve DNA from the sort of universe of DNA in GenBank. And then we could parse, remove redundancy, build gene trees, and exclude contamination and things like that. The dogs that are in primates and vice versa. So we end up with this big super matrix of 31 genes, only about 12% complete in the context of sites by species, but some genes are very well sampled. Some of the nuclear exons like VWF and GHR, some mitochondrial genes as well. And then other genes are less well sampled, but still have important information about longer branches. Sort of the way that we approached this was to, from the global supermatrix, first build a maximum likelihood tree, and then use that framework to divide up well-supported monophyletic groups, and then do separate Bayesian analyses where we could add in missing species with taxonomic constraints and get more accurate branch lengths in the context of substitutions. We then rescaled those patch clades to a backbone phylogeny that's estimated with fossils that has like kind of absolute time. And then in that rescaling, get and like stitching together, get the full tree. So it's sort of this multi-step process, because we can't estimate the full phylogeny in a time-scaled fashion on that many tips. Like our computational resources won't work for that at this point. Maybe in the future they will. This is more specifically what we actually did, right? So we had the global DNA RAXML tree. We had 28 patch clades with the DNA sampled and imputed species. So some species are, constrained to within a genus if another species does have DNA, and some are actually constrained at the family level. And then we had two backbones, right? We had a node-dated backbone and a tip-dated backbone where the fossils are actually included as sampled taxa, and then stitching it together into the full phylogeny. So the really cool thing about this is that we're able to capture uncertainty across the full mammal tree, not only in kind of temporal uncertainty, you see the node ages going back and forth, but also topological. So this, you see these balls kind of going back and forth. This is the main uncertainty between whether sloths and armadillos whether sloths and elephants are sister to each other relative to the rest of mammals, or vice versa. And so that this kind of core uncertainty at the base of placentals that is still largely an unanswered question. We didn't have to solve it. We just trusted what the data was saying, and we incorporate this uncertainty in the full sample of trees. And so when you do your analyses, you don't do your analysis on one phylogeny, you do your analysis on a sample of, say, 100 of those trees. And then you get that uncertainty in there. And sort of the goal here is to exclude alternative hypotheses, like given all that uncertainty, can we say that this event back here definitely did not influence the evolution of that group? In some cases we can, and then we learn something. So this is what that data looks like in the context of a lineage through time plot, right? So lineages on the log scale, time here. And so these red lineages are the 100 trees from the credible set of our analysis. The black is the consensus tree. And then these other colors here are sort of the previous mammal-wide phylogenies. You can see that they didn't have hardly any uncertainty in them. They were very definite about this is mammal evolution, but the data doesn't support that. Kind of breaking that down to these three orders of Rodentia, Chiroptera, and primates, and seeing sort of the different patterns in our study versus these other trees. And again, you can kind of see that there's this sort of weird, uneven uncertainty. For example, in this phylogeny, where there's no uncertainty in the base of the analysis, but there's more uncertainty towards the present, which is actually the opposite of what you would desire from a paleontological perspective. And yeah, so you can download these trees and subset them to different taxa. You just give it a species list and how many trees you want to download. So it's somewhat easy to get these trees to compare to your own or to do analyses. And this is sort of an example. So, pruning out a handful of, commonly studied organisms for biomedical studies, and you see, the node age uncertainty, but also, a little bit of topological uncertainty relative to the horse here. I think the horse will flip. Yeah, there it was. It's like, I think one tree out of 100 has a different relationship, but that, it's still important information to incorporate. And then we kind of, you step back and think about, there's all these diverse mammal ecologies and morphologies, from cetaceans to bats and selenodon and capybaras, obviously, tuco-tucos, really cool mammals. And, we want to try and understand that type of diversity in the context of what we know about their evolutionary timescale. So we end up with this, big time-scaled phylogeny, all in like a similar rate framework that we can then begin to investigate. And kind of first, I'll zoom into the tree, and actually I'll back up and say that what's mapped on the phylogeny here is these tip level speciation rates. And so these rates are actually calculated at the instantaneous present, but then they're shown just back on the branches just for plotting purposes, like just so you can see the patterns. But the deeper branches would have a different rate than at the tips. So zoom into primates here and kind of get the context for where humans fall in the phylogeny, which is often nice. 27 species in our superfamily here. And a really cool part of this analysis is that we were able to add in extinct hominids, right? So because of the ancient DNA genome work that's been done and published, and we have 4 members of Homo to kind of cut this long branch, which otherwise, go back to the chimpanzee bonobo branch. Yeah, so, quite interesting to have them in analysis as well. And then, thinking about, what can we learn from this perspective? And there's sort of new doors that end up being opened, right? So we have a modern perspective on the molecular rate framework that we can then compare to the fossil record. So our analysis using Bayesian macroevolutionary mixtures, BAM, kind of on 10 different phylogenies from our sample, and so showing sort of the variation across those 10 trees and the rate shifts relative to fossil duration information. So we did kind of an innovative thing in wanting to compare these pulled rates are basically when these purple shown here, pulled rates is this terminology for when there's no fossils in the analysis. So it's kind of pulled by missing extinction events. So this is what our phylogeny shows. And then we pushed the speciation rates by conditioning those speciation rates on the fossil extinction rates that we were able to calculate from those genus durations. And so when we do that, we see a big spike in speciation between the KPG mass extinction and the Paleocene-Eocene thermal maximum. Kind of both extinction and speciation was happening during this time, and it's completely absent from the mammal tree, which, it means that these lineages that speciated also went extinct and they didn't survive to the present, which is important. It's a bias in the tree. And so really kind of what we end up showing is that it's not until this overlap occurs towards the present, maybe the last 10 million years, that we can really trust the rates of speciation calculated from the mammal tree. So we missed this whole Paleocene between 66 million years ago and 56. This pulse in fossil turnover is missing from the extant time tree. But there's really, this reliable information starting from about 10 million years ago and at the present. And then, that sort of says, okay, well, maybe we can't really trust this knowledge about rate shifts deeper in the phylogeny, but we can definitely trust these last two, which are also the largest in magnitude. So let's take a closer look at those. And, really interestingly, the largest rate shift in the tree is Taxonomy's 𝐞-𝐣𝐚𝐦. And, win one for Southern and South America. This is really cool. Like what's going on with 𝐞-𝐣𝐚𝐦s that's so different? In particular, because the second largest rate shift is these Tropos flying foxes. And, you're like, whoa, these on the surface, maybe these are like the most different mammals you can imagine. The largest, flying bats to, small to medium size subterranean rodents, right? But they have this thing in common of, kind of gene flow or isolation in the context of their environment, right? So actual oceanic islands for Tropos flying foxes throughout the Indo-Pacific. And, you could say subterranean islands of these really fragmented, small geographic ranges, maybe easy to be isolated from one another in terms of populations that then go on to be species. And the bats, while they can go long distances, once they get to a place, are going to leave a population and another lineage might go forward, so that you get a similar type of dynamic. And then, we can kind of take a look at that in the context of ecological traits. So here's those rate shifts again plotted on the phylogeny. And really, we're saying that it's mostly these tip level rates, these tip DR, that are the most reliable. So let's look at the correlations with those, and in particular related to dispersal ability. So we calculated this, what we call a Vigility Index, that is largely allometric, right? So it scales with body size and home range size and geographic range size, and allows us to extrapolate across mammals. And you get, kind of large differences from artiodactyls and carnivorans, towards smaller bodied organisms, including, bats and rodents. So our index does not account for flying ability in bats. So that is one thing to consider. And we looked at two other traits as well, this diurnal activity, either daytime active, diurnal, or not. So just a binary. And you see that kind of showing up, including, a couple bats that are daytime active, and squirrels, obviously, primates, some, cetaceans and artiodactyls, and the latitudinal extent. So these kind of really interesting to see in a macroevolutionary or macroecological context. Like these are the lemurs shown here, right? They're like restricted to Madagascar, and you see like 𝐍𝐚𝐭𝐡𝐚𝐦 rodents here, and the tenrex. That's the Madagascar signature. It's really interesting. And then, actually doing an actual comparison, right? So we have the species level diversification rates, these TIP-DR, relative to vigility, and doing phylogenetic generalized least squares analysis, this PGLS, to correct for the influence of phylogeny. And while the actual trend line itself doesn't necessarily show much, these are consistently strong negative effects, right? So we have the highest recent speciation rates are shown in animals with the lowest vigility, the lowest dispersal ability, things like the tuco-tucos. And that seems to be consistent not only across mammals, but these different trophic levels as well. And so I think what this ends up sort of kind of fits in with some of these earlier ideas we were talking about, where speciation might be really rapid, but so is extinction. And so it's sort of this turnover-based idea where you have the extinction here and the speciation that are sort of chasing each other. And so you end up with a long branch with a bunch of species right at the tips. And that's sort of like, so something to kind of keep in mind in this context. Conversely, with diurnality, we think of this as more, access to new niches, right? So I think most ancestral mammals were probably nocturnal, and then there was different originations of daytime activity. And so we see a consistent positive effect of diurnality on those tip level rates, that, it's the fastest speciation rates are in those daytime active groups. This kind of, is putting it in the same framework, it sort of makes sense, right? That if you have access to more resources, your extinction rate is going to go down, and you have more persistence ultimately. And it can either be in the context of, also a change in speciation rate, or not. It can just be like these sort of low extinction, low speciation lineages, but some sort of key innovation might have occurred to lower the extinction rate in that group. But part of what's shown here in part A is that some of this is a theoretical necessity, right? So if you have high extinction rates, you have to have high speciation rates in order for us to even observe you, right? Like at the present, the only lineages that we'll be able to see as still extant will also have high speciation rates. This is like an impossible space here where you have high extinction and low speciation. You're just going to be gone. So this kind of makes sense. Lastly, we have the latitude dynamic, right? And so I've been sort of framing this in the context of time, right? Because the tropics are older than the temperate region. And quite interestingly, you'd expect there to be an effect, but at the tip level, there's no effect of latitude in our analysis. As repeated across 1,000 different phylogenies, a couple of trees show significance, but it's not a consistent relationship. And this is, really interesting when you then kind of zoom out and say, okay, let's look at this pattern over geography. So what's shown here is the tip level speciation rates as mapped using geographic range polygons at the present and overlaid with a grid. So it's sort of this macro ecological approach. And we see that there's a lot of high speciation rate cells in the north temperate region in particular, but also in the south temperate region in southern cone, and through into the Indo-Pacific here. So that the way this manifests, kind of comparing species richness versus this mean speciation rate is an inverse, right? It's sort of a mismatch, right? The places where you have the most species richness, you don't have the fastest recent rates. So it implies this sort of high turnover scenario that a lot of these species that are being originated in temperate environments are going to go extinct, or have been going extinct in the past, at least. It's hard to predict the future. I will mention that we can't just draw a line through this because the widespread species are overrepresented. They're in many cells, they're like multiple counted. And so you need to do a subsampling in order to draw a line through this. And that's sort of the next step of where we're going. We're also investigating a more mechanistic model where, we have north temperate, south temperate, and tropical, and like shared ranges across different species, and looking at the speciation, extinction, and dispersal rates across these different regions. So this is what would be a 5-state GeoHissy model. This is kind of where we're looking at it. And this is a collaboration with Daniel Caetano, who's actually now a professor at Towson University near New York. And then, just kind of zooming back, and then you see that there's a lot we can learn from this all mammal perspective, and a lot of additional questions that we haven't even thought of, and that you might investigate yourself. And I definitely encourage people to look at this data. But overall, we have sort of improved honesty of rate estimates. We can kind of actually now look at rate variation across lineages. The through time component is, it becomes less reliable the deeper back you go in the extant time tree. But really, we see this role of geographic isolation in driving recent species diversification, and this kind of, comparison with the fossil record, showing that the last 10 million years are the most reliable. And then I think, this is a novel insight kind of out of these ecological analyses we were doing, is that there's these two main modes of speciation, one in which a lot of extinction also occurs. And I would kind of put both the 𝒕nomies and the Tropos flying foxes in the same category of this turnover-based speciation. And that, this persistence-based speciation could be diurnal mammals, and perhaps things in the tropical region, globally, that have much lower extinction rates and older lineages, things like 𝐸𝒂𝒎𝒻 rodents, for example. But then, sort of zooming out and really putting this emphasis on, we need to be champions for taxonomy, because it is the language of biodiversity. It's not going away. It's our indexing system. And it's ultimately the key to understanding really important ecological interactions, including these mammal to pathogen dynamics that are increasingly important. And so, yeah, kind of with that, end up closing. This is my lab at Arizona State University now. Encourage you to contact me if you have questions or want to follow up about any of this. And just thank these people, and thank you all for your time. I can take some questions.
Thank you, Nate. Thank you so much for the nice talk. We have time to questions. Would you recite the questions in Spanish?
Yeah, no, in Spanish is okay.
Ok, perfecto. Muchas gracias. Comenzamos con las preguntas.
En español o en inglés o portugués.
Thank you. Hay unas preguntas. Pablo.
Nate,
Hola, Pablo.
¿Cómo va? Muy linda charla, realmente. Veía en tus imágenes que el número de especies de mamíferos que se van reconociendo a través del tiempo cada vez es mayor. ¿Qué proyección tienen de eso, digo, hacia el futuro? ¿Piensan que va a continuar esa tendencia? ¿Tienen una idea de hasta cuánto? O eso, básicamente.
Sí. ¿Qué número?
¿Cuál es el número final de mamíferos?
No sé. Actualmente, yo pienso que 8.000, 9.000 es posible. Cuando nos ponen una regresión, una línea en el papel 2018.
18.
Sí. Yo creo que fue 8.000.
8.000.
Más, pero no sé exactamente. Es su trabajo, Pablo.
Está bien. No, bien, pero digo, podemos pensar que en los años que siguen vamos a seguir describiendo especies. Todavía tenemos varias especies por describir.
Sí. ¿Cuánto cada año es posible para vos?
En Argentina, nosotros estamos describiendo en promedio una especie por año desde la década del 80. Aproximadamente es una por año, desde la década del 80, ¿no?
Sí.
Así que, digo, hay años que hay más, años que menos, pero es una en promedio.
Sí. Interessante. Eu, in inglês, un momento. I would encourage everyone when you publish a new species to definitely include a range map and to include like these are the DNA sequences that we know represent this species. Because and it's interesting too, and the third thing, a very clear statement of this is the not only the nomenclatural change, but the taxonomic change that I'm advocating in the paper. The problem is that we have lots of papers published that and we have to manually like look and see and try and understand what they meant and what the outcome is. But that is, the author of the paper at the time is the expert, right?
Okay.
And ideally, all that information would feed into, a common format into global databases.
Bien, muchas gracias, Nate.
Sí, gracias.